Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Zulfa Batrisha Miti Mustafa at 72467, and today I will present about the effect of ethylene hormone on the ripening of bananas. So I will start with the first part, which is the abstract. Our aim was to explore how natural ethylene produced by ripe bananas influenced the ripening of unripe ones. For the method, we placed unripe green bananas in sealed containers with ripe bananas to allow ethylene exposure. A control group of green bananas was kept separately. Over 4 days, we observed changes in color, texture, and aroma. The result shows that banana exposed to ethylene ripened faster. They turned yellow, softened, and emitted a sweet aroma earlier than the control group. Our conclusion supports the use of natural ethylene as an effective, eco-friendly, and low-cost method for post-harvest fruit management. It holds valuable application in both science and agriculture. Next is about the background of study. To give some context, banana are climacteric fruits, meaning they continue to ripen even after being harvested. This makes them ideal for observing ethylene's role. The banana was chosen for this project because its ripening stages are clearly visible and easy to monitor. It also gives a measurable response to ethylene. Ethylene plays a crucial role in ripening. It triggers metabolic changes, breaks down starch, degrades chlorophylls, and softens tissue. The production of ethylene in banana follows the autocatalytic trend, meaning once ripening starts, ethylene production increases and speeds up the process.
Next, let me briefly explain the significance of our study. Practically, this research helped farmers, sellers, and distributors reduce post-harvest losses. By understanding how ripening and storage work, we can extend fruit shelf life and reduce waste. It also promotes eco-friendly handling as we focus on natural, hormone-based ripening instead of chemical preservative. Furthermore, this can support local agriculture by strengthening supply chains and improving the quality and marketability of fruit. Academically, the study deepens our knowledge of plant hormones like ethylene, particularly how it affects ripening in climacteric fruits such as bananas. It also contributes to post-harvest biology by offering valuable data for better treatment methods, especially in tropical agriculture. Next, our main objective is to observe how natural ethylene produced by ripe bananas will influence the ripening of unripe bananas in a sealed setup. The scope of the study includes simple visual and sensory observation, like changes in color, texture, and aroma. We focus solely on natural ethylene, meaning no external chemicals or gas injections are involved. The study excludes any lab-based testing, synthetic agents, or nutrient analysis. It is a natural and low-tech approach aiming to understand fruit-to-fruit interaction through ethylene alone.
Next, we're gonna go with a literature review. This literature review, actually, we're gonna see about banana ripening, plant hormone, and ethylene biosynthesis. Okay, as a banana ripen, there are actually 3 key changes occur. First, starch is broken down into simple sugar, making the fruit taste sweeter. And second, the peel color change from green to yellow as chlorophyll degrade and carotenoid develop. Lastly, the texture soften due to enzyme like pectinase breaking down the cell wall. The main hormone behind all of this is actually ethylene. It's produced from the amino acid methionine through a pathway involving SAM and ACC. Ethylene acts as a ripening signal, and interestingly, it also promotes its own production in a feedback loop called autocatalysis. Other hormones also play a role, like auxin and MN1-MCP can delay ripening, while ABA support it. Ethylene works by binding to the receptor, which trigger ripening genes. Key enzymes involved include ACS, ACO for ethylene production, and expansins, and PGs for softening, and LOX for flavor and aroma development. In summary, banana ripening is a hormone-driven process, with ethylene playing a central role in making banana sweet, soft, and ready to eat. Next, for methodology, for this experiment, we actually use unripe banana of similar size and condition, all freshly sourced from a local market. Each banana was rinsed with clean water to remove any surface dirt. The treatment include two groups. The treatment group had one ripe banana sealed with one unripe banana inside a container, and a control group had only an unripe banana sealed alone. The goal in this experiment actually was to test if ethylene from the ripe banana could speed up the ripening process of unripe one. All banana were stored at room temperature and were observed them daily. We track key change such as color from green to yellow, texture or firmness, and aroma strength. We also took a note and photograph to monitor the ripening process closely. Okay, next for result for day 1, we actually have 4 type of experiment here. We have control, and we have 3 type of treatment. Treatment group 1, group 2, and group 3. Okay, for the day 1, control group banana was kept completely green and showed no sign of ripening. It had a firm texture and no smell was associated with it. For the treatment group, the peel tip was slightly yellow. However, the majority of the surface was still green and consistently was still solid and was no particular smell. That's all. Okay, next for the result for day 2, the control group, the change of color was slightly and certain places start to become light green and yellow, and the banana were not yet very soft and smell was weak and not existing. For a treatment group, there was more pronounced ripening. The peel was already of a distinct yellow color with some brown spot appearing. The texture was soft and faintly sweet smelling banana fragrance could be detected. As we can see from the image, the unripe banana also slightly become more yellowish because it's a day 2 treatment, obviously. Next, for day 3 result, for the control group, the banana was started to moderately ripen. There was some patchy yellowing on the peel and very little browning. The texture was a little softer compared to that one day 2, and it had a slightly sweet odor. For treatment group, banana was about 90% ripe. The peel was of a yellow color with a few brown spots. The texture was soft and there was a strong sweet aroma, which showed that was a considerable progress in the ripening process as a result of the exposure to ethylene. Okay. Next, for the day 4 control, day 4 result, for the control group, the banana highly ripened. There was a little patchy yellowing on the peel of this and tiny browning. In texture, it was a little softer than day 3 and had a sweet odor. For the treatment group, banana was left to ripe completely. The peel was yellow with some brown spot. It was soft in texture and possessed intense sweet aroma, which revealed that the ripen process had gone considerably ahead because of its long exposure to ethylene.
So next, I will be explaining the discussion for the method of this report, and our findings shows a very interesting result. That is, the ethylene speeds up the banana ripening more effectively compared to the natural processes. Take the treated group as an example that it shows the faster changes in color, texture, and also aroma even by day 2. Why? This is because the ethylene activates ripening genes and enzyme, which leads to the faster and more uniform in ripening. Meanwhile, the control group, which is the untreated banana, ripens slower due to the lack of external ethylene. And also, all of this prove that the ethylene is useful in the post-harvest storage to improve banana quality and consistency.
Here we go to the conclusion part. The results of this study demonstrate that ethylene significantly enhances the ripening process in bananas. Bananas exposed to natural ethylene exhibited earlier changes in color, texture, and aroma compared to the untreated crop, which is beneficial for both producers and consumers. These observations confirm ethylene's critical role in regulating ripening through metabolic and enzymatic activity, making it essential in controlling fruit development. The use of natural ethylene presents an efficient, low-cost, and environmentally friendly approach to post-harvest fruit management, which is especially useful in commercial agriculture and export industries. This method supports improved fruit quality, extended shelf life, and reduced post-harvest losses, allowing fruits to stay fresher during storage and transportation. Overall, the study underscores ethylene's value in advancing sustainable agricultural practices while ensuring better taste, texture, and less waste for consumers. In the future, this method can be applied to other climatic fruits, supporting precision farming and reducing unnecessary spoilage. These findings also open doors for more research into optimizing ethylene use across the fruit industry. Thank you. That's all from our group.